So let's go ahead and get started. To shamelessly plug myself and particularly my students, I needed some good news, and this was literally in my inbox this morning, that our manuscript was just accepted. So very excited about that. Second thing is that on the 17th of February, so that's a couple of weeks from now, on a Friday, the dean of the department, or sorry, the College of Liberal Arts and Sciences will be joining us. And I'll let you try and figure out who she is. Probably the one who doesn't have a clicker. I won't be pointing her out to you. The other thing, of course, is what's happening next Wednesday, our first midterm. What that means is we will have a review lecture on Monday. I think I'll still have some clicker questions there too, at least at the very beginning thereof. But there's not going to be any new material on Monday. All the material that will be on the exam will be what we covered today. So I won't be doing any more new stuff, and that's partly to give me a chance to go back and look at my notes and go, oh my God, I did a horrible job of explaining that the first time around, or something like that. It's sort of a way that I can save myself, as it were, in that whole process. Are there any questions at this point about the exam? I've posted old exams with and without keys. They should be available. Yes? What kind of Scantron do you need? The big blue ones. So those are the ones you need, not the little skinny ones. And the main reason for that is we got a new scanner in the office, and it doesn't take the little ones anymore. They're also way easier to scan so that I have copies of all of your exams. Exactly that one. Thank you. The blue one. Great. Any other questions before we get started? Okay, so again, today we'll basically start off where we left off last time, which was in sliding clamps. These are the processivity factors that are absolutely required. Oh, yeah, sure. Sorry. Pardon? Can you use note cards for the exam? No. I'm afraid not. Plus, how you would shrink everything into a note card, you'd like a huge microscope or something probably to see them. I'll take your word for it. So sliding clamps. These are what the polymerase needs to make sure that it stays on the DNA. And as it turns out, these sliding clamps are also really good at bringing other proteins and other protein complexes to the replication fork where they're needed. So it's not just a process that is interacting with the DNA polymerase. It's also helping bring together a lot of other proteins. One of my colleagues who works on these things thinks of kind of like a tool belt, like the replication tool belt. So it's got the DNA polymerase hanging off here, primase hanging off there, RNase H hanging off on the other side. So interacting with lots and lots of different proteins. Turns out that this particular structure, the ring around the DNA, is extremely similar in all kinds of different organisms. This is the one from E. coli. It's called the beta sliding clamp. There's also PCNA, again, more alphabet soup. Proliferating cell nuclear antigen is what PCNA stands for, proliferating cell nuclear antigen. The reason for that is that when you're proliferating, what are you doing? You're replicating your DNA. And as we'll see, you actually need many, many copies of this. So it's something that's present in a nucleus. You have many copies of it. That's the antigen the antibodies are going to be reacting with that interacts with it. And even viruses have very similar structures. This is the one from bacteriophage T4. And as was mentioned last time, I think, how do you get this ring around the DNA? You have to have a specific other protein complex to do this. The, again, incredibly creatively named clamp loader complex. So this is an ATPase, which will bind to one of these sliding clamps. Again, it's a ring, but at that point, it's not around the DNA. It opens up that ring structure, allows it to associate with the DNA, and then that ring structure closes along with the hydrolysis of ATP. And so that's what you need these clamp loaders for, is they're ATPases. They basically hold that clamp apart till it gets to the DNA, and then let it associate. And once it's on the DNA, it actually stays on the DNA for quite a long time. And how it comes off is an interesting question that no one really has a good answer to. So to risk belaboring the point here, this is a really nice image, I think, of the sliding clamp. In this case, it's the beta sliding clamp. It's only got 2 subunits. PCNA, as I forgot to mention, is actually 3 subunits, which is kind of interesting, even though they have almost exactly the same structure. One's in 3 and one's in 2. And fits really nicely around the DNA here in this space-fitting form, sticking basically into the major groove. And this really kind of rotates around the DNA in the major groove and holding it there. You had a question in the back. Yeah? This is for replication. So this is, we haven't gotten to transcription yet. That's like after the midterm, so don't even bother thinking about it for now. Except for the central dogma. No, but yeah, this is just for replication. It's just for interacting with the DNA polymerase. And to risk getting a little overboard here, it's a DNA-dependent DNA polymerase, which means that it binds to DNA, uses DNA as a template, and makes DNA. When we talk about transcription later, you'll see the differences that come out of that. And so, again, what this sliding clamp does is it gets loaded onto the DNA by the clamp loader, interacts with the DNA polymerase, and these DNA polymerases are not processive. They would love to just fall off the DNA and float around in the nucleus if we're a eukaryote or just in the bacterial cytoplasm. But since they're bound to the sliding clamp, they stay there, and they'll keep replicating until down here at the bottom, where the polymerase now hits one of these primers, and then the polymerase falls off. The sliding clamp stays on. And this is also where you get back to that tool belt approach. What do you need now to do once you hit one of these primers? It's RNA. What has to happen? You need to get rid of it. It needs to become DNA. And so you need RNase H, or in the case of E. coli, a DNA polymerase 1 that will get rid of this. And as we'll see in eukaryotes, there's also something called the flap endonuclease, which will get rid of that. Yeah? So pushing the DNA polymerase along? No, it's not pushing the DNA polymerase along. Actually, the DNA polymerase is translocating, i.e. moving along the DNA due to that hydrolysis that's happening. And so the DNA polymerase will actually bind to where there's a free 3' OH, and that keeps moving as soon as it keeps adding one. So it's actually the DNA polymerase just kind of pulling the ring along in this case. Okay, other questions about this? Good, no, when he says that, what does he mean? There'll be a clicker question, exactly. So sliding clamp seems to be necessary for genome replication because DNA polymerases are not processive, DNA polymerases have low fidelity, cannot associate with DNA without a clamp, hydrolyzes ATP, and it acts as a helicase. Oh, helicase. I hate autocorrect. That should be a helicase here. Helicase. Ten, 5, clutch. We seem to be pretty evenly divided between A and C. And I'm sorry about this overlap here. So it seems to be necessary for genome replication because DNA polymerases are not processive, low fidelity, cannot associate without a clamp, hydrolyzes ATP, and acts as a helicase, again, typo. No one likes the last two. The question is between the first three, basically. And the question here really has to do with what's meant by processive and what's meant by fidelity. What does processive mean? that it stays on and keeps adding nucleotides, right? Fidelity is what? Makes mistakes. Does the sliding clamp have anything to do with making mistakes? No. Absolutely none. Okay, so B's out. So we're down to binding to DNA or falling off of the DNA. So which one is it? It's falling off, so it's A. Question in the back, yes? So I'm not sure I quite follow your question. Sorry.
If you had a mismatched base pair,
yes.
as you're adding nucleotides to it, the plant is so specialized and small enough, would it pass over a mismatched base pair, or would it get stuck until that base pair?
Okay, yeah. So the question is, as the clamp is sliding along, would it basically recognize a structural problem that has happened in the DNA? Turns out that for mismatches, no. But when we talk about DNA damage after the midterm, yes. So some of both. And it has a lot to do with, again, the structure of the DNA. It's not so much the sequence, it's the actual structure. And clearly, mismatched base pairs are going to have a different structure. So it's that structure which is being detected. Yeah, sure. Yes, so DNA polymerase can associate very well without the clamp, and those were the original experiments that they did with E. coli, just purifying those individual polymerases, and they showed that they needed a template, they needed a primer, and nucleotide triphosphates, but that was it. You didn't need anything else. Now, to make sure it's processive and actually replicates the genome, then you have to have these sliding clamps. So question here is, would you see it without its clamp in a smaller organism? Okay, so I'm going to paraphrase a little bit here, pardon me. But the smaller organisms, the organismal genomes that we've talked about, even the smallest ones are a couple of 100,000. So it turns out, yes, you always do have a sliding clamp in those. We want to think viruses as organisms, which of course are the coolest things ever. Then in some cases it seems you don't, particularly for the ones with the very small genomes. But in some cases you do, and I showed that bacteriophage T4 sliding clamp, which was actually one of the first ones that people studied. That's a viral genome, but it's relatively large. It's a couple of 100,000 base pairs in length, so it probably has to do with that. Although in most cases the viruses are in cells that already have sliding clamps, they just use the cellular one. So, but as far as I know, there are no organisms that do not have a sliding clamp that aren't dependent on some other part of the other machinery. But it's a good question, and it turns out that the way people find these things, we'll get to in just a second when we talk about mismatch repair, has to do with the mutations and how you look at those. So what's present at a replication fork? We basically talked about all these things, but I wanted to emphasize what these things really look like, because it's a little confusing, and wrapping your brain around it, at least for me, was rather challenging to start with. So I want to spend a little while going over this. We've got our leading strand here. We've got our lagging strand over here. Leading strand is the one that's just going from 5' to 3'. You've got a polymerase. You've got a sliding clamp. This can just keep going and going and going as long as you have a single-stranded piece. How do you get the single-stranded piece? The activity of the DNA helicase, which is now wrapped around one strand instead of the two strands. But again, this loop structure. So that's pretty straightforward. On the other hand, you've got this lagging strand, which is being replicated semi-discontinuously, also, Okazaki fragments. What that really means is you've got to start doing replication many, many times on this lagging strand. And it turns out this is exactly what happens also at the origins of replication, that hopefully we'll get to at the end of the lecture today. You have a DNA primase, which again is an RNA polymerase, putting down these little blue RNA bits. Then you have a clamp loader, which is this green piece right here, together with a sliding clamp that's open. So as soon as you get this primer put down, now the sliding clamp can get associated with it. You can get a DNA polymerase that will now extend. Now, all DNA polymerases are going from 5' to 3'. None of them are going back the other way because of proofreading reasons. So how do you put all of this together in one complex? Basically, what this is they call it sort of a trombone model. And we'll look at this. We've got a great animation that we'll take a quick look at in just a second here. But basically what happens is once this primer is formed, then you can have a DNA polymerase that will go along it until it hits the next primer. Then this DNA polymerase will actually fall off, wait for the next clamp to be put down, and then extend from there. That also means you need a lot of the single-stranded binding protein, because all of the single-stranded binding protein is basically waiting for this polymerase right here to get to the end of that Okazaki fragment and then bounce back and take a look at it. Yeah?
Is it the same polymerase molecule, or is there just a lot of polymerases floating around waiting associated?
So the question is it just one polymerase molecule that's hanging out right there at the replication fork, or are there lots of DNA polymerase molecules floating around and then come in and associate? Turns out they're all together, and they're all stuck together. And people showed this really nicely early on in the process. You remember we talked about DNA polymerase 1 that Arthur Kornberg found? Well, the reason he found it was because it was the one that was the most common in the cell. Turns out it's DNA polymerase 3 in E. coli which does this. And that DNA polymerase 3 is at very low concentrations because you just have one leading strand, one lagging strand polymerase, but always associated with each other at the replication fork. And that actually is shown here in the blob up at the top here, but people have looked at leading strand and lagging strand. This is an electron micrograph where you have leading strand and lagging strand, and then this loop structure out here, and this is now your replication fork complex. Slight cartoon here. Again, polymerase, DNA polymerase 3, DNA polymerase 3, all connected together with these protein-protein interactions. And again, this is something we're going to talk about again and again and again through the course. Protein-protein interactions with a clamp loader and then a sliding clamp. And if you just purify these enzymes, these enzymatic activities, you'll find all of those things associated with each other. And so another way of looking at this is here. It's basically exactly what we just looked at, only mirror image, looking at it in the opposite orientation. This one's a little bit closer to what the real structure actually looks like. Again, primases, helicases, sliding clamps, and leading strand polymerase, lagging strand polymerase. Here it's got these single-stranded binding proteins bound here, should be bound over here on this side as well. What happens with a structure like this, this is an image from a paper, if you want to take a quick look at it, should be freely available, is when you have your lagging strand polymerase here gets to the primer for the next piece, this one at least has the single-stranded binding protein on it, it then will dissociate from the sliding clamp. You see it still stays attached right here. So that was your question about coming in from somewhere else. And now we'll move to the next sliding clamp, which with the clamp loader just put on the new primer, and extend from there. So what does this look like? This is one of my favorite videos for looking at, oh yeah, let's get rid of this wonderful post here. And take a look at these molecular visualizations of DNA. This first part is that part where we have nucleosome condensation. If you go a little bit further, now we'll look at the DNA replication process. Load up the full screen. So here is our complex. Right here, there's the lagging strand like this. Your leading strand is down here. There's your helicase pulling the strands apart. There's that clamp loader coming in from the back, putting down a sliding clamp. The purple is the polymerase. Right here. Lagging strand, so it's bouncing back and forth in this trombone, which is the template for that lagging strand, pulling out, get to the end, go and start again. Process it again and again. This is. I think it's worth taking a look at this. You're wondering how the process works. It's missing a couple of things.
Most obvious is flagging single-threaded DNA binding proteins. And then there are a few other things that we'll talk about in just a second. Yeah. Okay, so basically what you're asking is when is breeding happening? When is that backing up, exonuclease going on? So again, we're replicating from 5 to 3. What about the 3 to 5? Going back in the opposite direction. That's happening while this is moving forward. And so let's put it in the wrong nucleotide, take one step back and keep going. Yeah, in the back. Pardon? So the helicase, it's sort of right in the middle, but whether it's actually wrapped around the leading strand, you have to think about its template, then through the template, the lagging strand, pulling those two strands apart. To my knowledge, it's always a template for the lagging strand, but that I'm not completely sure about. Dr. Purcell, you would know. Yeah. Okay, so the question is basically, are those sliding clamps used again once they get in the Eukaryotic fragment? For DNA polymerase, no, because it's back doing its Eukaryotic fragment. But in terms of getting all of the enzymes that you need to get rid of that RNA primer, yes, it's still being used. And it may actually be the presence of one of these sliding clamps may also be a marker of the lagging strand as you're asking about in the back. In fact, the fact that you have sliding clamps associated with it, that means that that's a lagging strand versus being a leading strand. And that does turn out to be important for a couple of things here too. Okay, so let's I highly recommend taking a look at that video a couple of times, very least, to go through and take a look at it. So that was a very general sense. Unfortunately, we're talking about eukaryotes and bacteria, some extent archaea. Well, they've all got different names for things, which is really ridiculous, but this is what has happened. So we already talked about ECNA versus the sliding clamp. Of course, they nuclear antigens, so it can't be in bacteria. Nuclei. Then we have a loader, which is the RFC complex. Not DNA polymerase 3, because again, we're not E.coli. It's not DNA delta. There are about 8 or 9 different DNA polymerase in eukaryotic cells. But DNA polymerase delta, also DNA polymerase epsilon are the main ones which are involved in the replicative polymerases you have in eukaryotic cells. RPA is the single-stranded binding protein. We have a helicase. In this textbook, it's called the antigen, which is an interesting science. The antigen is actually where antigen from a viral infection. 40, which of course they first studied. It's actually now MCM, or the mini chromosomal maintenance protein, almost making some gins that we will talk about through the rest of the classes. And for a long time, people studied replication by looking at viruses and studying replication. That's where that comes from. It's a helicase, basically exactly the same thing. But as you'll notice here, up in the back, now they've got around the template for the leading strand rather than the lacking strand. And I can't remember exactly the details on how all of that is getting put together. So on the lagging strand, there are a couple of things that are a little bit different. Got our DNA primase, which is putting down your RNA primer. But in eukaryotes, you have extra DNA polymerase alpha. And basically all the DNA polymerase alphas do is extend primers a little bit more after the DNA primase. So this is something that doesn't exist in bacterial replication, just present in eukaryotic replication. Then at the end here, you end up at the end of your trying to get rid of your primers. There's RNase just like you have in E.coli. But there's also another thing, which is called FEN1 down here at the bottom. Now, FEN1 stands for flap endonuclease. We talked about exonuclease, you and DNA, right? Well, an endonuclease cuts in the middle of DNA. So it's an endonuclease that goes in the middle of your DNA. But what's it cutting into? It's cutting a flap. That flap is this RNA, which actually gets moved out of the way by the DNA polymerase coming through. So RNA flap, this extra piece that gets cut off. And so it's flap endonuclease is basically doing that extra job. There's also this topoisomerase out here at the end. I'll talk about those in just a second. So I find it's useful. This is the RNA's left primer that gets cut off. Basically kind of a redundant system. You've got RNA-H things, RNA hybrid, and the flap endonuclease is going to cut that off. Now, last time we talked about the really bizarre aspect of DNA polymerase 1, but as well as having a 3' exonuclease, also has a 5' exonuclease. So it chews up nucleotides in front of it. So it can also get rid of the RNA primers. But that's only present in bacteria. It's not present in yeast. Yeah. The question is, does DNA polymerase alpha extend the primer with DNA or RNA? It extends it with DNA. So it is in fact a true DNA polymerase, unlike DNA primase, which is RNA polymerase. Okay, so I find I'm over this in much detail, but I find it a really useful reference to try and remember all of the different key names. Again, the alphabet soup with all of these things. DNA polymerases, 3 delta, primase, anything ready is going to be different. All alpha, you don't have the equivalent. In bacteria, DNA polymerase 1, you don't have the equivalent in eukaryotic systems, FEN1, you don't have the equivalent in bacterial systems. So we'll put these things together. Any more questions? Good. No. Turn this back on. Which of the following proteins binds to single-stranded DNA? RPA, RC, NA, FEN1, RNase-H. Classic kind of question that would ask on an exam. Not that anything will be happening in the near future. And 5. Lift off. We have lift off. So RPA again. These are the kinds of things I would expect you to remember. Again, I'll give you that table on that slide. SSB, RPA. What's RFC? Clamp loader. DNA is the clamp. FEN1 is flap endonuclease. RNase-H is DNA hybrid. Yes. I would know the actual as well. Yes. It's also very confusing. I didn't exactly talk about what all these acronyms stand for. RPA, RFC, etc. Really want to know it's replication A. That's useful. And replication factor C. Again, totally not useful. I find DNA is somewhat useful because there's a whole bunch of it. And thinking about it from that point, FEN actually makes sense flap endonuclease. So I'll try and the ones that I think make some kind of sense. I'll talk about those that don't. We'll just use the acronyms. Yeah. question here is actually a very good question. Having to do with terminology is RPA a single-stranded binding protein? Yes, it is a single-stranded binding protein. But there's also SSB, E. coli, and they would just call it SSB. So single-stranded binding protein, E. coli, is another name for it when you're not going to get into it. Different from bacteria. But if you talk about SSB, that would go E. coli. RPA is going to be for eukaryotes. Yes. So the question is RPA have the same function with a different structure? And the answer is exactly yes. It turns out you might have seen there are actually three little pieces together in cartoons. So it turns out that the RPA is three different segments come together in terms of making that structure. And if you remember from last time, SSB is two come together. Does seem to be sort of a version of something that has to bind DNA. If you look at that high-resolution structure, then it turns out there are some similarities between the two, but in general they're pretty different. Yeah. So the question is basically paraphrase what happened to binding protein after you have replication. So you can't have those binding proteins there and still replicate the DNA on there. It turns out polymerase actually bumps them off. But they're still around. So they can bind back on as soon as the helicase opens up and they single-stranded DNA to bind to. Yeah. The single-stranded binding protein doesn't actually cover the base. Exactly right. But in order to have the polymerase coming along and the sliding clamp, it's not going to be available to be able to do that. So it's not actually binding the base per se. If it were binding base per se, it probably more like a base-specific binding, and you don't want that. You want something very general. But you need to have at least places in order to get the polymerization to happen. Okay, I need to select answer here. Otherwise, all these people who write will get points for it. Okay. So again, some may have noticed beginning that we're looking at the application forks. There was this funky thing off to the right, or left, called topoisomerase. It's an ACE. That means it's probably an enzyme. But what's it doing? So it's changing the topology. You got topology in math. I didn't know. So that makes at least 130 one of us. So what topoisomerase do is they change the topology of DNA. Why do you need to change the topology of DNA? Well, what happens when you replicate and you make single strands, particularly with helicases, you pull these strands apart. You're pulling the strands apart. You're untwisting them around each other. Well, if you're pulling them apart and the other end is stopped, what happens is that has to compensate for this untwist way. You're going to have to twist again in the opposite orientation. And we can look at that here. Here's the case of we've got our untwisted. I twisted a little bit, which is just running around here. That will then wrap together. And that's your supercoilin that's happening. We have extra things, extra wraps which are happening due to the unwinding process here. Now, if you don't have a topoisomerase change this overwrapping, then what happens is the polymerase actually gets stuck. Literally the helicase, because there's many of these things. It's been so overwhelmed. You can't get any more. And you get stuck. So what you need are topoisomerase to change the topology of your DNA. And so that's the reason you have these topoisomerases to take care of that. The one text basically exactly the same thing here. There are two flavors of topoisomerase. Again, incredibly creatively named Topo 1 and 2, but there's actually good reasons for that. Topoisomerase 1s cut one strand of DNA, and topoisomerase 2s cut two strands of DNA. And what that looks like is here. There are two possibilities for dealing with this kind of overwrapping, underwrapping. So you can just change one strand relative to the other. This is what happens with polymerase 1. Is you have a double strand of DNA, which gets cut through this type 1 DNA polymerase that uses an OH on a tyrosine side chain. Very typical reaction. That OH now is basically pretending to be the 3' DNA backbone. So it binds here. Now you have a free OH, and the DNA just wraps around itself. This is a reversible reaction. And so the OH change now the topoisomerase is no longer bound to DNA, and the DNA is reattached here. So what you're doing is you're cutting one of these strands, wrapping them around, putting it back together. So that's one way to deal with this overwrapping process. There's another way, which is topoisomerase 2. And so what topoisomerase 2 do is they take double-stranded DNA, cut both strands, pass another double-stranded DNA in between, and then put those strands back together. And again, mathematicians actually absolutely equivalent logically, and change these things back and forth. We're not going to talk more about linking numbers and look at old exams, linking numbers and back twist and drive. We're not going to get into that. It gets confusing. But these are equivalent. That's all we need to know. Is passing double strands through double strands. We're taking one strand, wrapping around are equivalent to each other. So these are the ways to deal with that extra binding that you get just because you're pulling the two strands apart. So you have to have topoisomerases, otherwise you can't continue to replicate. Yeah. So the question is here, basically, when you have a replication before topo 1 or 2 associated with it. And the answer is you have both. Some of you have topo 1, some of you have topo 2. And it seems to vary depending on different replication works. We'll see an example. We actually have to have a topo 2 in about 12 minutes. Assuming that I get there. So got a replication fork, leading ligand, etc. And proofreading, polymerase has its nice checking structure, but occasionally the wrong base gets put in. And we talked last time about the fidelity that you get just the DNA polymerase. And the fidelity you get just the DNA polymerase is about 1 in 10, 1 in 100 million. What's the general mutation rate? 1 in about a billion. How do you get one to the other? Turns out that there's this wonderful repair mechanism right after replication, which is why now rather than we talk about DNA repair a little later on. Called mismatch repair. And basically what this is it's another quality control system which says, hey, the wrong base got put in here. We need to take it out and put back in the correct base. Now usually these are actually normal bases. These C's and G's. They just get put in incorrectly. By mistake somehow, and quite often that's because of the polymerization that happens between amino versus amino groups are there, and the base pairs slightly differently. So how do we know about these things? Well, the first thing was that people looked at E.coli that didn't replicate well. Didn't make 1 billion mistakes. Made 1 in a couple of million mistakes. You can see that by looking at various different phenotypes of the organisms. But they said, okay, well, what's wrong with this particular one? Because again, geneticists loves to break things, figure out what they've broken. They just named all of these mutants based on the phenotypes that they saw. We'll talk much more about this on the course again. They have a way of trying to, at least for me, wrap my head around all these strange names. So they called all of these different mutants were mutants. So if you have a change in a gene, it causes a higher mutation rate. So what we ended up with mute S, mute L, lots of different genes, mute H, which are involved in this extra quality control process. We now know how they work, and that's down here at the bottom. There's some kind of mistake that happens. This gets recognized by the US protein. Here's the US protein, actually a high-resolution X-ray structure bound DNA. What's it recognizing? It's recognizing the structure of the long base. Recognizes the structure of the base. Then together with the protein, it will cut out a piece of DNA down here. That will then get remade and hopefully not put in the wrong base when this happens. So this is great, wonderful. But there's a big problem. There's a mismatch in your double-stranded DNA. How do you know which is the wrong one? They're normal nucleotides. So it turns out there are two generally different ways of dealing with this. E. coli smart. Probably has had more years of evolution with this. What E. coli does is after DNA has already been replicated, it puts methyl groups on. So here GHGC, sorry, it's a little bit hard to see behind your thing here. GHGC, all of the GHGC gets methylated, adenine after you've done replication. Does that well after you've done replication? So what that means is any strand that has a methyl group on it, that's an old one. That's the right information. You want to get the new strand, we put in the wrong nucleotide. So that's how E. coli mismatch system figures this out. This methylation versus one. On the other hand, eukaryotes don't do this. They methylate DNA, but for all kinds of different reasons. They have to do with silencing. Again, we'll talk about much later on. So what the eukaryotic system seem to do is they look for nicks. Now, what's a nick? A nick is a space in DNA where there's no covalent bond between the 3' OH and the 5' phosphate. This is what you see after you've gone rid of those primers. The RNase A and left nucleus. You've cut the polymerase come along. The associated DNA ligase goes together for that DNA ligase along. Then you have a space still in the DNA bank detected, and certainly now lagging strand. It's really obvious that that's where you have a problem. Leading strand is a little harder to understand exactly how the replication machinery knows that one of them is the original strand and the other one is not. Turns out that semi-discontinuous is not just the lagging strand. The leading strand is also somewhat semi-discontinuous as well. And so there are also nicks, new DNA made on the leading strand as well. So I've got nicks down at the bottom, H, HNCC, what that all mean. So H, again, these are names that have come from E. mutants, things that undergo very high mutation frequencies. This gene is mutated. UH, actually that thing that comes here in this strand step and gets rid of this extra DNA. In the eukaryotes, those things just be there because again of the lagging strand synthesis. HNPCC, what the heck is HNPCC? Hereditary non-polyposis colon cancer. Turns out that in this particular kind of colon cancer, one of the more common types of hereditary cancers, there's a mutation in one of the genes that's involved in this repair process. So just the fact you're missing a gene means that you're going to have much higher rates of cancer because your polymerase is not putting in the nucleotides appropriately. It's not correcting the ones that it's put in incorrectly. So these are very directly connected to each other. Yeah. Yes. So I'll just repeat this. But you're exactly right. So the new strand, the ones they put in, where you put in the mistake, because that's what you're trying to fix. That's the one that has an X in it. That's the one that gets taken away and resynthesized. It's easy, quote unquote, for E.coli is a strand that's not methylated. If you're a eukaryotic cell, you've got to see where those extra nicks are. The question here is, are those methylated restriction sites? That's way ahead of where they right now. It turns out, yes, they often are restriction sites, and that's protecting other DNA. We'll talk about that. Yeah. So the question is here, how many bases are actually removed in that process? Turns out there varies a lot. Probably less cut out in terms of E.coli. It's got this nice mechanism that it clearly what is stranded for one versus the other. Whereas if you're talking about Eukaryotic fragments, that can be much more challenging. Turns out Eukaryotic fragments in eukaryotic cells are shorter. As Eukaryotic you have bacteria. Probably because of some extent. And those nicks are there to serve as a way of being able to do this natural process. Yeah. Not sure completely follow your question, but you do actually need to, in a step here, just correct me if I'm answering your question here. But this here is just a bumped out piece, because the structure's wrong because you don't have appropriate base pairs. The backbone is completely normal at that. You have to have another nick that takes place. In fact, the bacteria, there are two nicks, and it's one here and one on the opposite side of this one. And that's what the H protein does that nicking. In order to give you cut on this side and a cut on that side of that red strand, which is the one you get rid of. You also need a helicase to get rid of that, and then it gets resynthesized. Yeah, but when you're in a mismatch, you're going to have a normal backbone because polymerase gone over that. Already got all your 5' and 3' linkage there. Okay. Quick review for all of these things. Which are the following hydrolyzes ATP? Topo 1, Topo 2, MUH. Okay. Number one versus Topo 2, MUH. Okay, so no one likes the mutes. Good, because they're associating with a mismatch. they're finding the right place. Some cases they're actually leaving the backbone. That's a hydrolysis reaction. But if you think about it from an entropic point of view, is hydrolysis going to be energetically good or bad? Actually, pretty good. Hydrolysis is pretty good to do. Certainly from a chemical point of view, it's that polymerization which is really hard. Forming the bonds, which is really hard. So none of those. Okay. Topo 1 versus Topo 2. Topo 1 is actually cutting and religating the DNA. But it's that OH provides what's in, and it just exchange reaction. You don't need any ATPase for that. So you do need Topo 2, which is literally pulling those two strands apart and putting them back together. So that's what you need ATP for. They want to finish up talking about initiation. This is where to start. There are lots of things that have to happen. You've got to put together that whole replication machinery. There ligand strands, plant loader, etc. But the main thing you need to do is get those strands apart. Because if you don't get those strands apart, primates can't do anything, and nothing else is a possibility. So what you need to do is pull those two strands apart. It really is that helicase, getting the helicase onto the DNA and pulling two strands apart. Once you've done that, you're going to get replication takes place at that position. In eukaryotes, there are literally hundreds of these things. In fact, thousands of them. But basically hundreds of nucleotides that are usually pretty rich in AT-base, high-rich pairs. Hydrogen, actually, it's easier to pull apart relative to each other. But to be able to figure out what these are, you need the so-called initiator proteins. Talk about in more detail later on when you do individual ones. And then once again you have this two strands pulled apart, your primer being put down. Then you can go to your elongating state, where your leading strand leading down here, lagging strand will start later, and you have processive replication. So what are these initiators? Basically, all that it is a binding protein that's going to bind to complex in some cases, to bind to origin and say, hey, this is the place where you want to start. And once it's done that, again, protein interactions. How many times protein interactions is, of course, hundreds. Rings in the other proteins which are necessary for pulling those two strands apart to the helicases, and then bringing in all of the other proteins which you need to get replication to start. Very well known in E.coli and yeast. Let's take a look at E.coli, the origin of replication, also known as C. It's the replication chromosome. C comes from, and it's literally where you start replication. E.coli has one origin of replication. One and only one origin, and then remember it's about 4.5 million base pairs. So takes off there. You have which initiates the two replication forks all the way around the genome, circular genome, and gets to the terminator, the opposite end of the genome, and these replicated genomes somehow come apart. To have the two replicated genomes apart, this is where you need a topoisomerase type 2. Because what ends up happening is as you replicate, you end up with circular DNA. They're actually interlinked with each other, kind of like chain nails. Two linked rings or a chain. You have to undo one, reattach it, so the cell divide, and each of them will have chromosomes that can go off and do their own thing. So you have to have this topoisomerase for separating them, but at the same time, you have 450,000 twists that you've done in terms of replicating. But also to be taken care of by Topo 1 or by Topo 2. Yeah. Yeah. So the question here basically is if you get rid of one of the orders of replication, can it actually use another one? In many cases, really no. It's very hard for E. coli to do that. On the other hand, eukaryotes are really good at using alternate origins of replication. Yeah. So the question is, why have two topoisomerases, topo1 and topo2? The answer is, don't ask why questions in biology. but probably it has to do with the fact that topo does require ATP, and you do need it for a couple of cases like here. At the very end of replication, topo1 does require ATP. So it's going to be less energetically demanding. One. So that's probably why. But whenever I say why, it's always going to have to be probably. Yeah. Again, the question is it or isn't it? The answer is it seems to be both. Getting back to the why question, as long as it works, it gets used. It doesn't have to necessarily be the optimal solution. Yeah. Okay, so the question is how does this help for mitosis? This is E. coli. Actually, it's not going through the cell cycle that you think about eukaryotic cell, but it does turn out that this cross-linking between the two is rather important. Talk about that later on. Yeah. Okay, sorry. At the beginning of that question, the question is different concentrations of topo1 and topo2. I'm going to hammer on that. Partly seems to depend on your growth phase, where you are actually growing. From organism to different, and there's actually a lot of topo1s and a lot of topo2s. So it's really hard to separate these two relative to each other. So the answer is it varies. Yeah. The situation where topo1 is specifically necessary. In theory, you can get away with topo2, but again, you need ATP hydroxylase for topo2. So probably, and a lot of things people do is they use mutations again, like I'm talking about these phenotypes, and so trying to remember what exactly the phenotypes are of some of those stuff. I'll check on that. Okay, so. We need to get to eukaryotic initiation. So we need to move through a couple more of these. Basically, the process here is now going through what proteins are involved in this process. Unfortunately, it's called DNA, C, again, not very useful, but the initiator, DNA A, this is an E. coli, binds to HC-rich sequences and forms a structure, a DNA protein structure, which now allows this most important protein, helicase loader. I had a question in the back of the last time about how do you get that helicase around that template strand. Again, it's a loader. Again, it's an ATP process. Well, open up the loop, form a circular form of your DNA helicase, and clamp it down onto the DNA. Once those two things get apart, you've got DNA primase, etc. Once you get started, you're good to go. So it's really all about this initiator protein and then protein interactions with your clamp loader. DNA is what happens in E. coli. The incredibly creatively named origin recognition complex in eukaryotes, or So what do these things actually look like? Some really nice structures now of where you have the DNA at the origin bound to different proteins. So work. You have eukaryotic systems. Turns out A is a very similar one. DNA A. You look here, these protein structures are really different relative to each other, but they're still serving the same role, finding that origin, protein interacting with it. Some viral proteins, even more different relative to this, but they'll do exactly the same thing. So it's a case of immediate transport apart. You have to have an initiator protein there. Then once that's found, you're good to go. licensing. So I mentioned before, you were talking about this too. In E.coli, you've got only one origin replication. That's because you only have the sequences DNA combined to one genome. That's great. It's wonderful. But it's that one place in the genome. Once you've replicated that place, you've got copies of it. What do you do now? Does DNA bind to that again and recruit the helicase loader, start again? That could be a real problem because you've got all these different replication intermediates going on. So how does E.coli again know that it's you that particular origin of replication? This gets back exactly the same way that it knows which is the old strand. After you're done with replication, what do? Methylate. You've methylated. Now you have those strands which are methylated. Once they separated, you only got what's called hemi-methylated form. That's because one of the strands is methylated, the other strand is not methylated. And so that's how E.coli knows that it doesn't bind to that says DNA is only going to associate with completely methylated DNA. It won't associate with hemi-methylated DNA. No DNA association, no helicase loader, no helicase, no replication. So that's bacteria. What happens in eukaryotes? Eukaryotes have a lot of problems, other than the present. Eukaryotic polymerases are slow. The genomes are really big. How do you deal with it? The only way to deal with it is to have lots of origins of replication. And so these are all called autonomous replication sequences, yeast. And basically, they're present all throughout the genome. They seem to be not terribly sequence-specific, and that means you use a lot of different ones and get away with it. Quite often, what you see are a bunch of origins of replication close to each other, and a big gap, and then another set. The way you can see this by looking at DNA being replicated. You can do this by using radioactive nucleotides and looking at where that DNA is actually made. And in fact, in that process, you can see that you have lots of different origins of replication. So E.coli, one origin of replication. You do it in a cell, and a eukaryote, multiple origins of replication. There are other ways of looking at that. Not going to get into. Skip to talk about your lecture, then on the exam. I just wanted to talk about the last step here, which has to do with the actual initiation of replication in any one of the particular origins of replication. Mentioned before, the origin recognition complex. This is your initiator. This is where you actually end up starting. So to finish up right here, you've got double-stranded DNA and origin that gets recognized by origin recognition complex. Then you have these two funky proteins that come in, bind this origin recognition complex, CD66 and CD1. CD stands for cell division cycle. And if you have a mutation in one of these, you have problems in your cell cycle. So that's where all those naming comes from. So CD66 and CD1 are the licensing factors. They're basically the equivalent methylations you have in your E.coli DNA. Because what happens is these guys get put together. You get your helicase loaded with your MCM. Then you have helicase activity starts here, but it's this phosphorylation step that happens right here. Phosphorylation happens to the ORC complex, but also to CD66. What's doing this phosphorylation? It's cycle-independent kinase. So the cell cycle, how is the cell cycle driven? It's driven through phosphorylation. And that phosphorylation is due to these cycle-independent kinases. Cycle-independent kinases will only recognize this initiation complex if you have 6061 and then that puts the helicase together. When you have phosphorylation, phosphorylate equals, which says, hey, you can't associate with C66 if you want anymore. And C66 is phosphorylated, which ends up leading to degradation through ubiquitination, E1, E2, etc. So that's what we'll get to on midterm question runs away. So polyubiquitination, yes, does usually lead to degradation, and that's exactly what happens in this case. See you on Monday. Have a good weekend.